Hi, I'm James. I'm happy to share to you today about tumor microenvironment TME heterogeneity and how that impacts the response to combined thermoablation with immunotherapy. The TME differs significantly due to differences in various immune cell populations and profiles, and that then translates into a difference in immunotherapy efficacy, which depends on the availability of immune cell populations. We then hypothesized that high-intensity focused ultrasound HIFU can augment immunotherapy efficacy by increasing tumor-infiltrating immune cells. To answer this hypothesis, we start with using single-cell RNA sequencing to profile treatment-naive TMEs, specifically comparing between MT4 pancreatic cancer tumor model with NDL breast cancer tumor model. We found that in the MT4 tumor models, there were more B cells, neutrophils, with fewer CD8 T cells, macrophages, and on the right-hand side, pre-HIFU histology, there were dense stroma and sparse cells. In contrast, in the NDL TME, there were more CD8 T cells, NK, and macrophages with fewer B cells and neutrophils shown on the single-cell RNA sequencing data. And on the right-hand side, pre-HIFU histology, there were higher cell density. After high-intensity focused ultrasound, there was an increase in infiltration of cells in the ablated region. In blood cytokine analysis, after high-intensity focused ultrasound, we found that in both NDL and MT4, there was an increase in inflammatory cytokines such as IL-6 when compared to no treatment controls. We want to compare a two-component combination therapy system in both pancreatic MT4 tumors and breast NDL tumors. In this case, we collected the tumor 3 days after the treatment and used bulk RNA sequencing to characterize the responses. We then used digital cytometry to deconvolve the bulk RNA sequencing signal into cellular signals of possible origins. We found that in both pancreatic MT4 tumors and breast cancer NDL tumors, there was an increase in myeloid macrophage monocytes, CD4 B cells, and Tregs in the treated tumors. In contrast, in the breast cancer tumor only, there was dendritic cell activation in the treated tumor, but not in the contralateral tumor, and this is not seen in the MT4 pancreatic cancer tumor models. Dendritic cell activation is important in stimulating an adaptive immune response against the tumors. We then asked the question whether multi-component therapy can increase dendritic cell activation, which is important for adaptive immune therapy. In this case, in pancreatic tumor models, we combine ablation with CTLA-4, anti-PD-1, both checkpoint inhibitors, and anti-CD40. We found that in the ablation with CP4 in the contralateral tumor site, there was an increased upregulation in adaptive, innate, and TLR pathway signature genes that was stronger than the systemic CP4 administration. On the right-hand side, in the CyberSort digital cytometry comparisons, we found that ablation with CP4 in both the treated and the distant side increased dendritic cell activation shown on the top right-hand side corner, B cells, NK cells, and CD4. The combination of ablation with CP4 is translated into an increased survival benefit over CP4 only or no treatment control, as well as ablation and or ablation plus PD-1. This survival benefit is weakly correlated to the dendritic cell activation, NK and CD4 immune cells. In conclusion, we've shown that TME immune cell population affects immunotherapy efficacy with single-cell sequencing data. combining HIFU with PD-1 induces infiltration at the treated tumor. But if we compare HIFU with PD-1, CTLA-4, and CD40, and increased infiltration at treated and distant tumor. The transcriptomic responses of HIFU with CP4 is greater than immunotherapy alone, and the survival is increased and attributed to a combination of myeloid remodeling and infiltration of lymphocytes. I'd like to thank my lab for their immense support and Kathy for a lot of her guidances. I'd like to also thank a funding agency at NIH. I'd be happy to take any questions. Thank you for your time.